Hi everyone, welcome to all. This is Vidyam Academy's most expected question series for medical NEET examination. Today's topic is 12th Biology, chapter number 5, Molecular Basis of Inheritance. And into this subtopic is RNA world and DNA replication. And now it's being presented by Rajiv Patel. Once again, let's welcome to all. Let's get started. The first question is, the RNA world hypothesis proposes that RNA preceded both DNA and proteins as the primary biological molecule. Which feature of RNA best supports this hypothesis? So here the options are: option A, RNA is double-stranded like DNA and can store genetic information more stably. Option B, RNA can act as both an information storing molecule like DNA and catalytic molecule like enzymes, making it self-sufficient for early life. Option C, RNA uses the same 4 bases as DNA and is therefore older than DNA. Option D, RNA is found in all living organisms while DNA is absent in viruses. So what is the right answer for this question? Would you guess the right answer for this question? Yes. The right answer is option B. Let's see how and why option B is the right answer, the question which number 1 demands for. The RNA world hypothesis proposes that early life was based entirely on RNA before DNA, and protein-based systems evolved. The strongest argument for this is that RNA is the only known biological molecule that is both informational. It can store genetic sequence information in its nucleotide RNA splicing, just like protein enzymes. This dual role means an RNA molecule could theoretically replicate itself and catalyze its own biochemical reactions without needing proteins or DNA. Evidence includes the fact that the catalytic core of the ribosome, which makes all proteins, is RNA. So not protein, suggesting proteins were made by RNA before proteins could make themselves. That's what RNA can act as both an information storing molecule like DNA and catalytic molecule like enzymes, making it self-sufficient for early life. That's what answer B is correct answer here. Let's move to question number 2. Meselson and Stahl 1958 proved that DNA replication is semi-conservative. What did their experiment involve and what result confirmed semi-conservative replication? Option A, they used 32P and 35S labels. After one round of replication, all DNA was fully light, proving conservative replication. Option B, they grew equally in a 15N medium, then switched to 14N. After one generation, all DNA was intermediate density hybrid, confirming semi-conservative replication. Option C, they showed that DNA is synthesized in the 3 to 5 direction and both stand simultaneously. Option D, they labeled the parental DNA with 14C and showed the daughter DNA contained no 14C after 2 rounds of replication. So what is the right answer? Let's check whether you are right or wrong. Option B is right answer again here. Let's see why and how option B is correctly and right answer here. Meselson and Stahl grew equally for many generations in medium containing 15N, famously known as heavy nitrogen, making all cellular DNA uniformly heavy. They then transformed the bacteria to 14N, famously known as light nitrogen, so medium, and collected DNA after each generation, separating it by density gradient centrifugation. After one generation, all DNA formed a single band at intermediate density, hybrid DNA containing one 15N strand old and one 14N strand new. After 2 generations, 2 bands appeared, one hybrid intermediate, and one fully late. This pattern matched the prediction of semi-conservative replication exactly. Each parental strand serves as template, and each daughter double helix has one old and one new strand. Conservative replication would give one heavy and one light band after one generation. So dispersive would give one intermediate band at all generations. Only semi-conservative matched the actual data. That's what option B is the right answer. Question number 2 demands for. Let's move to question number 3. Which enzyme is responsible for synthesizing new DNA strands during replication, and what is critical limitation of this enzyme that requires another enzyme to initiate synthesis? Option A. RNA polymerase synthesizes DNA and can start synthesize on any template independently. Option B. DNA polymerase 3 synthesizes new DNA strands but cannot start a new chain. It can only add nucleotide to an existing 3OH, requiring a primase-made RNA a primer to begin. Option C. DNA ligase synthesis new DNA strands in both 5 to 3 and 3 to 5 direction. Option D. Helicase synthesis new DNA strands after unwinding the double helix. What is the answer? The correct answer is option B. Why? Let's check it. DNA polymerase 3 is the main replicative enzyme in prokaryotes. It adds deoxyribose one at a time to the 3OH end of growing chain, reading the template strand in the 3 to 5 direction while synthesizing the new strand in the 5 to 3 direction. Its critical limitation is that it cannot start a new DNA chain on its own. It can only extend a short RNA about 10 to 13 nucleotides, complementary to the template, providing the free 3OH that DNA polymerase 3 needs to begin adding DNA nucleotides. After replication, the RNA primers are removed by DNA polymerase, which has 5 to 3 direction exonuclease activity. The gaps are filled by DNA polymerase 1 and the nicks are sealed by DNA ligase. That's what. So here option B is right answer. Next, move to question number 4. In DNA replication, one new strand is synthesized continuously leading strand and the other discontinuously lagging strand. Why is the lagging strand synthesized discontinuously? Option A. The lagging strand template has a different base composition that slows down DNA polymerase. Option B. DNA polymerase can only synthesize in the 5 to 3 direction, but the lagging strand template runs 5 to 3 away from the replication fork. So synthesis occurs in short fragments, Okazaki fragments, toward the fork. Option C. The lagging strand is synthesized first and then unwound to allow leading strand synthesis. Option D. The lagging strand uses a different types of DNA polymerase that can only work in short bursts. What is the answer? The correct answer is option B. Why and how? Let's see the explanation. DNA polymerase can only add nucleotides to the 3OH end of the strand, meaning it only can synthesize in the 5 to 3 direction, never in the 3 to 5 direction. At the replication fork, helicase unwinds the double helix, exposing both template strands. One template runs 3 to 5 in the direction of fork movement. DNA polymerase reads this and synthesizes continuously in a fork movement. DNA polymerase must work away from the fork to read this template in the 3 to 5 direction, synthesizing short segments, Okazaki fragments, each 100 to 1000 nucleotides along in eukaryotes in the 5 to 3 direction. These fragments are later joined by DNA ligase after RNA primers are removed and gaps filled, creating the continuous lagging strand. That's what, again, option B is the right answer. Next, move to question number 5. DNA replication begins at specific sites on the chromosome. Which of the following correctly describes the origin of replication and replication forks? Option A. Replication begins at random sites and proceeds in one direction from each site. Option B. In E. coli, there is a single origin of replication. The two strands are separated and two replication of forks form, moving in opposite directions. Bidirectional replication. Option C. In eukaryotes, there is only one origin of replication per chromosome, making replication very slow. Option D. Replication forks move unidirectionally from the origin and rejoin at the termination site. What is the right answer? Could you guess the right answer? The right answer for this question is option B. DNA replication begins at specific sequence called origins of replication. In E. coli, there is a single origin. Proteins recognize this site, bind and unwind the double helix locally. So this is the explanation. Two replication forks form at this origin and move in opposite direction around the circular chromosome. This bidirectional replication ensures the entire genome is replicated efficiently from a single starting point. Eukaryotic chromosomes are much larger and linear, so they have multiple origins of replication, 1,000 chromosomes. That 5 simultaneous are in a coordinated sequence, so drastically reducing the total time needed for genome-wide replication. Each phase of replication proceeding from one origin forms a replication bubble. When adjacent bubbles meet, the forks merge and replication of the segment is complete. That's what, again, option B is the right answer. Next, question number 6. Which of the following correctly matches each enzyme to its specific role in DNA replication? Option A. Helicase seal sticks in DNA, ligase unwinds the double helix, DNA polymerase 1 removes RNA primers. Option B. Helicase unwinds and separates the two DNA strands at the replication fork, so primase synthesize short RNA primers. DNA polymerase 3 extends the new DNA strand. Option C. Topoisomerase synthesize new DNA primase, joins Okazaki fragments. Helicase adds nucleotide to the 3-OH end. Option 4. DNA polymerase 1 main replicative enzyme. In prokaryotes, ligase synthesize RNA primers. Helicase reads the template strand. What is the right answer? Could you guess the correct answer? Option B is right answer. Let's see why and how. DNA replication requires a team of enzymes working in coordinated sequence. Helicase uses ATP to break hydrogen bonds between base pairs and unwind the double helix ahead of the replication fork, separating the two strands to expose the templates. So primase, a specialized RNA polymerase, synthesizes short RNA primers, so 5 to 3 complementary to each template strand, providing the 3-OH terminus that DNA polymerase requires to begin synthesis. DNA polymerase 3 is the main replicative enzyme in E. coli. It with very high fidelity. Additionally, topoisomerase relieves the torsional stress ahead of the fork. DNA polymerase 1 removes RNA primers and fills the gaps with the DNA, and DNA ligase seals the remaining nicks to form a continuous strand. That's what option B is the right answer. Next, move to question number 7. DNA replication has an extremely low error rate, approximately 1 mistake per 10 9 nucleotides added. Which mechanism primarily accounts for this high fidelity? Option A. DNA polymerase randomly samples all 4 nucleotides and selects the correct one by trial and error. Option B. DNA polymerase has 3 to 5 exonuclease proofreading activity. It detects and excises incorrectly incorporated nucleotides immediately after adding them. Option C, the high specificity of hydrogen bond formation between base phase alone accounts for the fidelity without any need for a proofreading. Option D, repair enzymes separately scan all newly synthesized DNA after replication is complete and fix all errors. What is the right answer? Option B is right answer again. Let's see why and how. While the complementary base pairing rules AT and GC ensure that the correct nucleotide is chosen most of the time, this alone gives an error rate about 1 per 10 to 4, 10 to 5, not sufficient for faithful inheritance. The primary mechanism that reduces errors further is the 3 to 5 exonuclease proofreading activity of DNA polymerase 3 immediately after adding each nucleotide. The enzyme checks whether a correct base pair has formed. If the newly added nucleotide is incorrect, the mismatched base pair distorts the geometry of the enzyme's active site. The polymerase then switches to its exonuclease domain, clips out of the wrong nucleotide in the 3 to 5 direction, and resynthesis that position correctly. This real-time proofreading brings the error rate down 1 per 10 to 7. Additional post-replication mismatch repair systems reduce it further to approximately. Next, move to question number 8. Consider the following statements about the RNA world hypothesis and ribozymes. Statement 1. Ribozymes are RNA molecules that possess catalytic activity. They can catalyze biochemical reactions just like protein enzymes. Statement 2. The discovery of ribozymes by Thomas Zech and Sidney Altman, Nobel Prize winners in 1989, provided strong evidence for the RNA world hypothesis. Statement 3. In the rhizome, the peptide transferase activity responsible for forming a peptide bonds between amino acids is carried out by ribosomal proteins, not by RNA. So here are the options. Option A, all 3 statements are correct. Option B, only statements 1 and 2 are correct. Option C, only statement 1 is correct. Option D, all 3 statements are incorrect. Would you guess the right answer for this question? Yes, statement 1 and 2 are correct. Statement 3 is false. That's what answer, and option B is the right answer. Let's see why and how. Statement 1 is correct because ribozymes are catalytic RNA molecules. The first discovered was cell splicing introns in a tetrahymena. So Zech 1982 and the RNA component of RNase P. Altman. Statement 2 is correct because Zech and Altman shared the 1989 Nobel Prize in Chemistry for discovering ribozymes. And this discovery fundamentally supported the RNA world idea that RNA preceded proteins as the biochemical catalyst in early life. Statement 3 is also false because the peptidal transferase activity of the ribosome, which catalyzes the peptide bond between adjacent amino acids during translation, is carried out by the 23S RNA, so in prokaryotes, in 28S in eukaryotes, not by the ribosomal proteins. Ribosomal proteins stabilize the RNA structure, but the catalysis is RNA mediated. This ribosome as ribosome, a fact, is itself one of the strongest arguments for the RNA world hypothesis. Let's move to question number 9. So here the assertion A, DNA replication requires an RNA primer to initiate synthesis on each template strand, but the primer must be removed and replaced with the DNA before replication is complete. So here the reason are for this assertion. DNA polymerase 3 cannot start a polynucleotide chain from scratch. It can only add nucleotides to a pre-existing 3OH group. Once synthesis is initiated by the RNA primer, DNA polymerase 1 removes the primer using its 5 to 3X nuclease activity and fills the gap with DNA, and ligase seals the nick. Option A, both A and R are true, and R is the correct explanation of A. So B, both A and R are true, but R is not the correct explanation of A. And C, A is true, but R is false. And D, A is false. What is the right answer? The right answer for this question is option A. Both A and R are true, and R is the correct explanation of A. Let's see. So here assertion A is true because every new DNA strand begins with an RNA primer synthesized by primase, and every primer must be removed and replaced with DNA before replication is finished. Because RNA nucleotides in the final product would cause instability and eventual breakdown. Reason R is also true and gives the complete mechanistic explanation. DNA polymerase 3 lacks the ability to initiate a new chain. It cannot form the first bond without a pre-existing 3-OH, which is why primase is essential. After DNA polymerase 3 extends from the primer. DNA polymerase 1 uses its 5 to 3 exonuclease activity to degrade the RNA primer in the forward direction while simultaneously filling the gap with DNA nucleotides using its 5 to 3 polymerase activity, a NEET translation mechanism. Finally, DNA ligase seals the remaining single strand break between 3-OH of the new DNA and the 5-phosphate of the adjacent DNA fragment. So R completely explains why A is true. The primer is needed to start synthesis but removed to ensure a pure DNA product. And final question of this topic. Question number 10. Arrange the following molecular events of DNA replication or the replication forks in correct chronological order. P. Helicase unwinds the double helix separating the two template strands at the origin. Q. SSB single strand binding protein stabilize the separated strands and prevent re-annealing. R. Primase synthesis a short RNA primer on each template strand. S. DNA polymerase 3 extends the primer adding DNTPs in the 5 to 3 direction. T. DNA polymerase 1 removes the RNA primer and fills the gap with DNA. And U. DNA ligase seals the nick, joining adjacent DNA fragments into a continuous strand. Options A. PQRSTU, B. RPQSTU, C. PRQSTU, and D. QPRSUT. What is the right answer? The final question. Option A, PQRSTU is exactly right answer as per the sequence of a practical. Let's see the explanation. Replication at the fork follows a stick molecule sequence helicase PX first. It uses ATP hydrolysis to break hydrogen bonds and unwind the double helix at the origin, creating single-stranded template regions ahead of the fork. Single-strand binding proteins Q immediately coat the newly exposed single-stranded DNA, so preventing the complementary strands from re-annealing and protecting them for nucleoside degradation. So primase R then synthesis a short RNA primer complementary to each template strand, providing the crucial 3-OH that the next enzyme requires. DNA polymerase 3S binds to the primer 3-OH and rapidly extends the new DNA strand by adding deoxyribose complementary to the template. The leading strand continuously and the lagging stack of Okazaki fragments DNA polymerase 1 then displaces and degrades each RNA primer using 5 to 3 exonuclease activity while simultaneously filling the gap with DNA. So finally DNA ligase forms a phosphodized bond between 3-OH of the newly filled region and the 5-phosphate of the adjacent fragment, sealing the nick of completing continuous DNA strand. That's what option A is rightly matched, is the exact sequence. That's what option A is the right answer. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you, and thank you so much to all.